So I'll now give you a little bit of a demo of how to use BCILAB. And I'll start with an overview of the graphical user interface and how to use that. And I'll try to transition in time to the scripting part and show you a few of the plugins just so you get a taste of what this is about. After you've downloaded BCILAB from, say, FTP, what you get is a directory like this one here. This is actually directly downloaded from the internet. So you have, I just narrate this because it might be a little bit tiny to see. You have a code directory, which contains all the code. A dependencies directory, which contains all the externally maintained dependencies. And a user data directory, which contains sample data and so on. We'll process some of this. And a user scripts directory, which contains a few tutorial scripts primarily. But this would be where you might dump all your scripts. And one M file, which is bcilab.m, that's the main startup function of this toolbox. Also, in this build directory, there's actually compiled versions. So if you want to run this without MATLAB, you could go there. Importantly, there's release notes. So you're highly encouraged to actually read these release notes when you get the toolbox. It points to some extra places of documentation and updated, perhaps, internet addresses and things like that. What I'll do is I'll just start MATLAB now to and load the toolbox. So OK, so the first thing that that you will basically do is close some of this stuff is cd change the directory into this folder where you extracted it, and then you start the toolbox. Just typing BCILAB. It's basically also the same way in which EGLab is being started. So I'll do this, and then it'll start up. It'll take a few seconds. It's going to be a bit faster on the second try because it's all already cached. And you should be getting, you should be getting a little GUI here. So that's the main graphical user interface of the toolbox. And I'll actually walk you through this from left to right, basically. So fundamentally, this GUI allows you to do essentially everything that I basically described earlier in the previous lecture, so all the different processes and so on. The first question that you probably ask yourself is, how do I get some data into the toolbox so that I can process it? And that starts in the data source department, basically. So this is where you can load recordings and so on. And we'll do this here right away. Let's say there's a few tutorial slides on the internet also, which mostly mirror what I'm doing here now. So there's different tutorial data sets, and one of them is an ERP data set. It's event-related potentials, and it includes the flanker task that I described in one of the previous lectures. So that's a subject being presented with errors, and some of them are flankers that are triggering subject errors. There's two data sets in here. It's brain products format, and the two come from identical twins. I start with the second data set here. So after I've loaded this, by the way, it's the .vhdr file that you would need to open if you did that. It asks me if I want to sort of prune the data that I'm loading. So you can do that if you don't have enough memory or things like that. And I'm not going to do any of that. I basically just click OK. But before I do this, fundamentally, there's always a help button for every dialog. And it should take you to the documentation. The documentation is rather extensive. So load set has a little description text, inputs, outputs, and so on. There's tons of cross-references usually to other functions and so on. Like here, something about chain log inference and so on. Although it apparently takes MATLAB a while to load that documentation page. And so that's the help button. And then there's a few other optional features that you can work with such as how a trigger channel should be interpreted or so. But we're not going to look at that right now. I just say OK. Oh, great, and here comes my documentation. So now I've basically loaded a data set. If I, in MATLAB, typed whos, it would show me what variables are there. And there's something that's called last data. It actually is not the actual data. It's just a description of the data. So if I look at that, this is what I just said. The toolbox is a bit lazy, but that's fine. We don't want to tinker with this data just yet. So now we have data. The next step would be to do some analysis. And the first step in any analysis is, as I said, to decide on what kind of computational approach you want to run. So what algorithm do I want to apply, what BCI paradigm? And that's, you can basically rattle this off from top to bottom if you want to do an actual analysis. And by the way, there's icons down here. They are basically just the most frequently used steps from the menu. So I say I'm creating a new approach. And this brings up this dialog. What you can do is you can select which BCI paradigm you want to use. So as I said, you are never starting from 0. You're basically always starting with some existing something, like in this case, a paradigm template. And so what we want to do here is with this data, we have this flanker task data. There's a person making frequent errors. It would be an interesting question if you can predict whether the person is going to make an error by the time he presses this button. And so that's an ERP process, event-related potentials, and that's why we need an event-related potentials method. There's several in here, and one of them is this windowed means approach that you saw in one of the lectures, and that is also in the methods teaser. There's, again, quite a bit of documentation here. Let's see what the help thing brings up. OK, it gives you kind of an overview of the different paradigms that are there. And so after I've settled with this, I can click OK and start customizing this. In almost no case is the paradigm already perfectly tuned to your task and your problem and whatever. But there's always defaults for everything. So there's a default sampling rate, default time window that you want to look at, default frequency filter. The defaults come usually from some existing experiment or paper where this method was first introduced. So that's actually also in the help, I think. So if I bring up the help for this, somewhere down here is a citation for a paper that probably first proposed these particular defaults for some task. What we want to do for this flanker task is the following. We have a person pressing a button. And after that button, we think he's getting some kind of a cognitive response. And we might choose a time window of about, say, a second or so, in which we inspect the data after the button press to decide whether he made an error or not. And so I might put in a general range of interest in epoch for this method, which starts at 0 and ends at 1 second. Although I want to perhaps include a little bit of data before the actual button press to have some sort of baseline. So I start at minus 0.2. And I'll go a little bit shorter, say 0.8. Because after all, after the guy presses a button, you have to see this much data until you can make the decision. So 800 milliseconds is already quite a bit. Frequency filter, I'll go up to 15 Hertz. And then the next step is this windowed means approach asks you to dial in little sub-windows in the epoch where you want to where you want to average the signal to extract features. And I have no serious prior knowledge on this data, say. All I know is probably there's not much going to happen before 250 milliseconds after you press a button or so. Or 200, say. So I start at 0.2. And I end, I also don't know much about the resolution in there, say. I just go up to 0.25. So now I have one time window. That's not going to be enough. I want to basically cover the majority of this epoch with a few time windows. But I don't want to use too many because that would be too many parameters to learn, sort of the so-called curse of dimensionality that LDA is so plagued by, which is our default classifier. So I just use 5 or 6. So I go on 0.25 to 0.3. So even though this method happens to have hand-tuned parameters, you can usually just go on and type in some random ad hoc values, and it's still doing reasonably well, let's say. So 0.3 to 0.35. I'm separating this with semicolons. So this is MATLAB syntax here. It's basically a matrix. It has 2 columns and a couple of rows. So I go 0.35 to 0.4, 0.4, 0.45, 0.45, 0.5, and then maybe a little longer at the very end where we think there might be some positivity from the literature, which I'm not going to review now, 0.5 to 0.6. So hoping that I have no typo in here, I double-check this. If something doesn't work, especially in the tutorials, you basically know that you have a typo somewhere. So I typed this in, and now I'm done. Now, as you see, this is actually very few parameters. This method should have many more parameters. And this is because when you directly click the OK button in the previous dialog, you get a simplified mini configuration dialog, which is this one. The only thing else that I can set here is which classifier I want to use, which machine learning method, basically. I stick to the default linear discriminant analysis and say OK. I will show you in a second how you can massively customize this using a different button. Now I have defined my approach, and I can go on and use it. In fact, this dialog will put this into the workspace as a variable. It will call it last approach by default. I can also name it. I could edit the documentation if I did something inventive here, and I could go on and save it on disk if I wanted to. I just save it, so you can use it later on, my approach. OK, but I'm not going to use what I've just saved. And then I say OK, and I'm done. My approach is now completely specified, basically. The next thing that needs to happen is I want to continue my offline analysis. Now I basically want to calibrate or train a model using the data that I just loaded and according to the approach that I just defined. So that's the button Train New Model here. I go on that. So we're working gradually towards something that we can actually apply online, say in a human-computer interface system or so, or something that we can start interpreting. And when I do this, it gives me this dialog here. And here I can choose which approach I want to use, which data I want to use. So if I have multiple, I can select here. Then I'm asked to type in how the string markers in the data set relate to the labels that it should predict. So that's important. Do I want to have it predict 1 or -1 or 0 or 1? And what are the markers? There's probably dozens of markers in this recording, and only some of them are actually relevant. So I can click this Inspect Data button here and actually look it up to remind myself. But usually you will want to have this documented somewhere, of course. So I actually, OK. So there's a bunch of markers here. There's boundary markers, whatever. Importantly, there's this S201, S102, and so on. And as I'm looking at this, I just remember how this was specified. There's 2 markers for errors and 2 markers for non-errors. And it's left-hand errors, right-hand errors, and left-hand no errors, right-hand no errors. So basically, there's 2 groups of markers. And each group defines a class, an output value, or a desired outcome of the BCI, or desired output. So this is a special grammar here, or syntax, in which you type in markers in marker groups. By default, it'll just take the first string and assign it a 1. The next string, it'll assign a 2, and so on as a value. So this could be my first string. This could be my next marker string or so. This would be mapped onto 1. This would be mapped onto 2. What I want is I want to have subgroups. And this is with nested cell arrays. This is actually, there's documentation about this if you want to read up the details. So basically, if I had 3 classes, I could say, OK, my first marker, say, my marker 1, assuming that it's in a data set, then my marker 2 or so. And if I have more classes, I would go on and basically essentially say which are the relevant markers. And now if if I had multiple markers that were basically class 2 or user condition 2 or something like that, I would put them in a group. And this is done with these curly brackets. So I say my marker 2A, for example, and my marker 2B. What I've now defined is the following. I said there's 3 classes. The first one is indicated by the marker called my marker 1. The second one is indicated by my marker 2. And the third class, actually I have 4 classes, sorry, I'll get rid of this. And the third class is indicated by either my marker 2A or my marker 2B here, which are in a group. So, but to end this little excursion here, I just type in the groups that we have in this experiment. We have S101, S102, and S201 and S202. OK. So, and now I can basically, I'm done with the specification. The same line here is also, by the way, in the script. So you can directly copy this. So this is my markers. And then I can go on, say how I want this to be evaluated and so on. So for example, I want to measure mean square error or something like that, or area under curve. But what I actually want is I just stick to the default, which is going to be misclassification rate. You can decide whether you want a cross-validation or not. I want that. I use 5-fold. By default, it's a blockwise cross-validation, so it's not randomized, because it comes from a time series. And that's all. The last things, I'll leave those at the defaults, but I have the option to save the model under a different name and save the statistics under a different name. And again, there's a good amount of help. I just go on and say OK. And now it should start crunching numbers, hoping that I didn't put in a typo or something like that. So let's see how far this goes. It'll take a few seconds to compute, because it needs to do a cross-validation and so on. And especially if you have little memory, it might not be able to store, say, multiple data sets in memory. So it needs to unload it, load it again, and process it again, and so on. So that might take longer. But here we are. So a result just came out. What we get is some kind of a summary of the expected performance of this model. It's giving us an error rate. It's, in this case, 5%. So it's wrong 5% of the time. And it's broken down into true, negatives, false, negatives, true, positive, false, positives, and so on. You could export this. I just say OK. So this works pretty well. This means this particular model on this particular task is wrong 5% of the time. The chance level is, I think, here 25%, because person made an error on average 25% of the time. If you always say the person made no error, you're right 75% of the time. Anyway, so we have a rather well-performing model. Next thing is to actually take a look at it and visualize it, probably. Say if you're writing a paper, that would be the thing that goes into your paper. So here's a little visualization of this. In fact, it should be close to the same that we already saw in one of the earlier lectures. What this is the linear weights of the classifier assigned to the different features. And so there's, it looks like 64 features for the first time window, 1 per channel, and so on. Second time window, etc. So what you get here are spatial filters. So it's not easy to interpret those. For example, this here is the surface Laplacian. It subtracts the surrounding channels from 1. This probably picks up a dipole. And it probably picks up something that's right underneath, but it's not always as simple, especially if it's a checkerboard pattern like this one. Also, the weightings are all the same, so we basically know that the first window is particularly relevant. It's highly weighted. And the third window is also relevant. And so one is probably the negativity, and the other is positivity or so. And then there's a bunch of other windows where perhaps there's so much noise in the data that they are not all that important. There's, in theory, ways to localize these things better. So you can work with independent components and so on. You can do a whole bunch of things. There's a couple of papers on that. I point to some of those at the end on how to properly localize that, if you want to interpret it. Of course, if you just want to run things, you don't really care about the interpretations. So we have this model. We have an estimate of how it works. That's already good enough for, say, a results section in a paper if you have enough subjects. But what we want to do next is perhaps test it on some new data. And so we first have to load some new data. And what we'll do is we'll load the data from the twin. So I'll load the first data set here. Click. Now we have it. I actually overwrote the old data set because I used the same name, but it doesn't really matter. If we look into the workspace now at this point, we see there is an approach. There is the data set that I just loaded, the model, and the statistics. And you can inspect these various data structures and get the parameters out and so on if you want to. But we are not going to care about this. We're just trying to apply the model to some new data. And there's something here. It's called apply model to data. So I do that. I can select which data set, which model, which loss metric. And I stick to the default, which is going to be a misclassification rate in this case. I save the results in last results and say OK. And so now we're predicting for every instance where for every trial in the recording that was done with the twin, whether the twin likely made an error on each of these trials. If we do this, we see we get an error rate of 10%. So it's actually quite a bit worse, which is not really surprising because, among others, their cortex is folded differently and so on. The cap was probably positioned differently. And timing, I don't know if that is going to be different. So it is, however, still not too bad. So you're right 90% of the time. ChanceLab is probably still 25% here. So that's actually a pretty neat result. Now that's all offline results. By the way, you can always go back to the last results here with this review results button and plot them again if that's of interest, and export them from there and so on. The next step really is to go online. Now, actually, I didn't bring an amplifier or so today. So what we'll do is we'll take this data set from the twin and play it back in the background. And then we classify it online and visualize the outputs of the classifier. So all that happens in the online department here. And there's one section on where to get the data from, read input from. And I'll pick as a data source the data set source. So there I can select which data set to read from. And by default, it just uses the last one that I loaded. So OK. Now, actually, if I look into the workspace, first it needs to do a little bit of reprocessing and load it and so on. So now if I look at the workspace, what we have is a new data structure here. It's called last stream. And if I update this, you see there's something changing here. And that's just data being accumulated into this data structure. Actually, it just shows how many samples it has seen so far. This happens in the background. And so now I can say, OK, I want to run this thing and actually send the output somewhere. Run the classifier, basically, in the model. So I say, OK, I want to send it somewhere. I send it to a MATLAB visualization, just for fun. And I could have sent it over TCP to my stimulus presentation program or so. But I'm using this. And what you can select is which of the models that you might have or not have you want to use, which of the streams you want to use as data source, how you want to plot it, what you want to plot. I'm going to plot the distribution, probability distribution, and click on that. So now what we see is the following. We see a bar diagram that indicates the probability that the person is in condition 1, which is no error, or that he is in the error condition. Since errors are rather infrequent, the second bar should only infrequently spike up, basically, and the first bar should be way more likely. But there's one thing here, and this model was trained relative to certain events in this experiment. In fact, right off the top of my head, I don't even know if it's button presses or stimulus presentations. I will have to look this up. But what's important is this particular model, most likely, it has only ever seen data that is exactly at this particular latency relative to this kind of event. So if you always query it, the BCI, relative to this event, you will approximate, this in real time, this accuracy of, say, or error rate of 10% error. But if you slide it over data where all sorts of things are happening, such as a person is resting or a person is sneezing or so, you might get erratic behavior. And so it's important that you train the data on the kind of stuff that you want to later on test it on. Anyway, so I close this. If you are doing work with actual hardware, you will probably almost always use the lab streaming layer as input source. Except that now it tries to draw the bar diagram. I have to first stop my online processing. OK. So, so if I go into this lab streaming layer, here I can pick where I want to get the data from, using LSL and so on. So I could say I want an EEG stream on the network, or maybe I want a mocap stream, and so on. The only thing is that I currently don't have any such device at hand. But it basically, I'm just pressing Control-C, so that's why you get this error here. That's how it would basically work. So this is how to do some very, very basic, very quick analysis. You could go on if you have more data sets and do a batch analysis now. Run batch analysis. What it'll do is it'll automatically list things that are in memory. So it lists the approach here. It lists the one and only data set here. And it lists the markers that I typed in. And that's sort of most of it. I could go on and type in more approaches if I have more. My other approach, for example. And then it'll compare both approaches, basically, and produce the results and save them somewhere. For the data sets, I could say something like c:/test/**set. So it'll use every data set in this folder and run the analysis on that and stuff like that. You can have separate testing sets. But I can't really explain all the features of this in this little tour. So there's some documentation on how to run this method. Also, just real quick, you can essentially really customize the entire pipeline from this little dialog. So you can customize how you want to load your data, such as things like, I want to enforce something in how my trigger channels are extracted or something like that. You can customize how you want to train. So this is kind of a small version of the training dialog. So I can say I want to use, say, area under curve or something like that as metric. See where this is going here. You can say how you want to predict on a new data set. So you could say I want to predict estimate the expected value of my person's cognitive state or something like that. And you can say where you want to save it and things like that. So it's pretty flexible. And also you can use, obviously, the same function also from scripting. I'll show you that. So it's a rather efficient way of running over a whole collection of data sets without clicking too much. If you want to change settings, there's the settings area here. You can, for example, say where your data lives and where your results are supposed to be stored and things like that. You can configure how you want to, and if you want to, cache intermediate results. So for example, I could say I want to store intermediate results in C, directly in drive C or so, which is not a great idea, and things like that. Although it can be somewhat counterintuitive if you're caching intermediate results. Just a quick note. It saves you a lot of time, because if you want to calculate something again that you had calculated before, you get the result immediately. But the trouble is that if it's possible to make mistakes with that. So say, for example, you overwrite your original data on disk and expect your results to change if you rerun your whole pipeline. But if they were stored and cached, you suddenly get the same results again, and so on. So since there is more state involved and scattered over your disk, it can be counterintuitive unless you really are principled in how you work. And so that's basically most of the stuff that's in the GUI. I'll just show you two more things. The help. So the help is very organized. So there's a menu where you can say, OK, I want to read something about the filters. And then you get this list. There are all the different filters, say the IIR filter, and documentation, and so on. And there's help about scripting and stuff like that. But what's basically the next step is if you want to really go beyond the built-in methods that come with this toolbox, when you want to customize methods heavily, or perhaps create your own. And we'll start with the heavy customization. What I'll do is I'll go back and load the old recording again. And I'll edit my approach. So actually, I might even start over, but I'm not going to start over. I'm going to take the approach that I have and use this thing here, which is called review/edit approach. And what this brings up is a kind of a panel of all the parameters that this has, and parameters that are not really utilized. So fundamentally, what you see here is sort of some top-level structure, and this is signal processing, which is all the things that you can do in the signal processing department, so to speak. This is the online pipeline that you can configure here. So you could say something like, I don't want to use the spectral selection filter here. I want to disable that. Instead, I want to use an FIR filter. And so I enable it, and I say, I go to frequencies and type and say what frequency I want, such as 0.2 to 0.5. It's actually 4 numbers in this case. It's sort of lower and higher transition band. 10, 15. I could say I want a linear phase filter, minimum phase, or whatever. And then you can say, OK, I also want to remove AOG artifacts, I artifacts. This is a regression technique. So I turn this on. And I could decide which channels I want to use as I artifact reference channels and so on. Or say, oh, I also want independent component analysis. So I'm operating on components, and I want to use Infomax. And then you have the Infomax parameters here, and so on. So it really goes very, very deep. You can go on and say, I want to clean artifacts in this way. And when I remove bad channels, I want to use this and that parameter to determine how much I want to clean or so. So all of this is kind of hidden in this dialog. What I, however, do at the end of the day now is I'll turn off some of these fancy bells and whistles. So for example, I don't have I channels in my data. I'll just stick with this FIR filter. And the only thing that I'm going to change is in the prediction department, which determines how I extract features and how I do machine learning, I'll change the classifier from LDA. I'll change it to, say, to a logistic regression. This is logreg. It's basically using a different plugin. And then I'm going to change which flavor of logistic regression I want to use. There's a bunch of variants, variational base, and so on and so on. I use one which is sparse. So it uses only few features. And as a result, it'll only use a few channels and a few time points. So I just set this and say OK. And I won't save it on disk. I just save it in the workspace. So now I have a new approach. It's pretty custom, sparse, classifier, and all that. By the way, the plugins that I was just turning on are in this code directory, somewhere in this folder here. And specifically under filters, I use filtFIR here. And under machine learning, I was using ML train logreg and ML predict logreg here. You could look at the code and read the documentation, and I'll show you some plugins by the end of this. But before we really dive into the plugins, I'll just train a model with this approach, reusing all the same defaults that we just had. And now it'll crunch probably a little bit longer because the logistic regression is a little bit slower than LDA. And I hope that I don't run out of memory, actually. So my laptop has only 4 gigabytes. That's already sort of at the low end of what you want to do with BCILAB. ideally you run on some kind of Linux server or so. Now, to run things online, you don't need as much memory. It's just when you want to train and cross-validate and parameter search and so on. So this worked. What's interesting is that it's somewhat worse. It's 11% error. OK. But what might be interesting is actually to visualize this model. What you see now is, I'll show you the color bar. 0 is green, so it uses only very few channels now. In particular, it only uses a few channels up front here, and something here, and whatever, probably an artifact channel. So basically, you could remove all these other time windows, and you can remove all the other channels, and run this whole approach with just something like 5 channels, and get your 11% error or so. So that's actually not too bad. And now I could actually put this to the test and run it online. So I say again, I want to read from a data set. I want to write the output to a visualization. The mouse is flicking out on me. And so here's the output of this classifier. What's actually interesting is it seems to be a little bit less erratic. If you look at this, the other class doesn't shoot up as frequently. Also, logistic regression is one of the few really proper probabilistic classifiers. LDA isn't really, the output is the distance from the hyperplane, so to speak. Logistic regression is giving you something that you can actually interpret as a probability. Anyway, and now I could go on and do a comparison of this method and the other in my batch analysis or things like that. So it's basically the same story if you use any other paradigm. So for example, you could use this paradigm DAL ERP, which is the very advanced ERP analysis, but it would run much longer. We definitely don't have the time to go through this now. But I just say one thing, and this is, if you don't even want to see this simplified configuration dialog, you can directly say, when you create a new approach, full edit when done. And I'll do this for DAL ERP, sort of. And so then you get directly to this review panel here. So you don't have to deal with the basics. All the stuff that shows up in the GUI is harvested from disk. So if you put in a new function somewhere that you just coded, it'll show up here. And you can immediately configure it and use it with existing or new methods. And also all the help and so on comes from there. What we should do next is I should show you something about the scripting. So I'll show you in a script that's equivalent to what we just did. And there's a tutorial script. It's called tutorial_ERP1. So I just type edit tutorial ERP1. It's in the user scripts folder. So this is the script equivalent of our analysis. It's actually, so it comes with a little bit of documentation. The first thing that it does is it defines some markers. It defines the time windows that I typed in earlier. It loads the data. This is one line, basically, IO load set in this case. Then comes this part where we define the approach. And so we typed in a bunch of things like 0 seconds to 0.8 or whatever, some spectral bands. It's usually a one-liner to define an approach. Although you might obviously kind of split it up across multiple lines if it gets a bit too complex, like I'm doing it here. What you generally say is there's some grammar to that, and I have slides on that. I explain this in a little bit more detail, in somewhat better detail. You generally say what paradigm you want to start with. It's this here, window means. And then you can go on and override sort of all the nested parameters. So you say, oh, in the signal processing department, under epoch extraction, I want to use something different. And under spectral selection, I want to use something different. In the prediction department, and specifically feature extraction, I want to use the time windows that I just defined earlier, and so on. So it usually boils down to just setting a bunch of parameters and choosing which paradigm you want to use. And then there's just two more lines. One trains the model, and the other visualizes it. The train the model line calls the function BCITrain, which is doing this calibration and all that. All it gets is the data, the approach variable, and the markers. the marker specification. So I should be able to run these lines here. But before I do this, I actually clear my workspace so I'm not wasting too much memory. And I just run, actually I run this cell here, including the visualization. And this should now actually reproduce the visualization that we saw in the beginning, more or less. So at the end of the day, it's rather easy to write up an analysis in the scripting, and you can easily whip up 10 different approaches which take up 3 lines each or so, and compare all of them, for example. So here's the other visualization again. And the script shows a few more things. I'm not going to narrate all that, but here is testing this on some new data. This is running this on the twin again. So there's one function, it's called BCI predict. It gets the model, the data, and it outputs the statistics and all that. And so here I'm just plotting it, test misclassification rate 11% and so on. And then there's an online part to the script. You see, it basically covers everything that we just did in the GUI. It reads the data set from the background, in the background. It sends the output to a visualization, and then it just waits until I press a button. So I do this. Here we go. And I click a button, and so now it's over. And you could have used other plugins here, like run read biosemi to read from a biosemi amp, run write TCP to send it over TCP to some program, and things like that. And here is actually also the sparse classifier use case. So define some markers, define some time windows. This is the same story. Load the data, define the approach. And here, what's different is in the prediction department, under machine learning, specifically the learner function, I use logistic regression, and I override one variable here, one parameter, the variant. I set it to VB art. ART stands for automatic relevance determination. And then you train the model. This is the same line. This is the same line. So running this should give us a sparse result. And the last chunk shows just a few more. This is quite interesting, I think, for people who really want to explore the toolbox. Shows various methods and designs and so on ERPs, for example, with support vector machines, with group sparse, classification and regularization. Here's the sparse classifier again, just for confirmation. It uses also this advanced low-rank dull ERP approach that I teased you about. And it tries some wavelet-based method, and so on and so on. So you can easily write this all up in just 10 or so lines. And here's a loop that rattles this all off. Also, there's another tutorial script. I'm not going to run this, but it's this tutorial_batch, which shows how to use a batch analysis. So that's the whole script. It basically says, here's my time window, here's my marker specification. That's my training data, my evaluation data. And these are asterisk expressions. So this is going over all data sets from a particular BCI competition. Then I define 3 methods here using different paradigms, actually. And at the end, I am running this batch analysis. And this goes over all data sets and all methods and compares this and so on. So here you see how the script version of this batch analysis dialog is spelled out. And so that concludes the intro to the scripting. Now, since we haven't really discussed oscillatory processes yet, I cannot yet show this to you. I'll show this later. There's tutorials for oscillatory processes also. This is tutorial ERD1, ERD2, and so on. They go across a whole range of methods. That's certainly very interesting for people who want to see and want to have some code that actually runs, where they can drop in their own data and then use a whole menu of different methods. This is probably on the order of 25 different methods or so. Stationary subspace analysis, whatever. But what I'll show you last to finish this part is I'll show you some plugins, what they look like and how they're written. And as I said, all the plugins are somewhere in this code directory. And signal processing plugins are in the filters directory there, subdirectory. So we might start with something really simple. Let's start with filter FFT. It takes an epoch signal and outputs an epoch signal. And for each epoch, it does an FFT. So here's the function. It's a single MATLAB function, has some documentation. It outputs a signal, and it gets some varg in. OK. There is some boilerplate here, which we ignore for now. Here's the actual code. So here it does an FFT on signal.data. It removes some stuff here. And it actually has an option, which is whether you want to have logarithm of the power or not. Actually, it calculates the power. So it's a spectral transform. And so that's in the, it's very simple, very straightforward AOG style code. And the only thing that's different is you have some extra stuff. This exp begin fun line and the exp end fun line. You always put these in. It's basically part of the contract. Every filter has to have these two lines. And what they basically do is they allow you to automatically get a description of the filter pipelines and produce these lazily evaluated expressions and so on. And the other thing is there's properties associated with these filters in this case. Say, for example, they have a name. This one is called epoch FFT. And so you specify these things. Or, for example, this filter, it makes no sense to run this before you've extracted epochs. Because, I mean, you can, in theory, run an FFT on a whole data set. But this particular filter is defined to depend on the function that extracts epochs. And there's a few more things that are being said about it, such as it usually follows a couple other filters here. And so what these basically just express is ordering relationships between filters. So this one says, I want to be run after set makeup was runs, and also preferably after filter project and filter window. And thanks to these ordering constraints, the toolbox can generate this GUI and put them in some sensible initial order. So if I start with a new approach or so, like this one, I get everything in some meaningful order. For example, resampling comes first, because after that, everything uses much lower sampling rate, is faster, and so on, and things like that. So that's basically resolved from these definitions. But you can override it if you want to. And you can also not run it as a filter chain, but in parallel if you also want to. So that's what a simple plugin looks like. I'll show you another one. The one that removes AOG by regression. So it's basically the same story, except this one is a stateful filter. So it outputs a signal in a state, some documentation. There's some standard AOG style code in here. Here's the actual mathematics, some recursive least squares calculation. It has the exp end fun again, exp begin fun. It declares some properties. And this method has a series of parameters. You see them actually in the GUI. So if I make a new approach again, under AOG removal, if I click this, I have a bunch of parameters. I have, for example, forget factor here. It's by default 1. Or I've removed AOG, which is a checkbox. And there's documentation about that, which is down here. So remove AOG has a parameter that says remove the AOG channels after processing or so. So some minimal documentation. And all of this is automatically generated from this part here, which is sort of the main, you could say, magic of the BCILAB plugin system, which allows you to say, I have an argument. It's called AOG channels. It's empty by default. And here's the documentation string. And I have another one. It's called forget factor, default 0.995. And it has this documentation. And in total, there's 5 arguments or so. With this little specification, you have something that can generate dialogues and that can generate stuff that's somewhere hidden in the GUI. And so for this reason, you never, ever build GUIs in this toolbox. But people get GUIs for free. And that's really important if you just want to write the math and someone else just wants to use a GUI. That's sort of two worlds colliding. But here, you can easily bridge that. Other plugins are other plugin types are similar. So machine learning methods, for example, say ML train SVM perf, which is a particular SVM implementation, is a function. It outputs a model, has a whole bunch of parameters, a bunch of references. It defines a lot of arguments. It defines basically all the arguments that the SVM perf toolbox supports. You know, which kernel do I want to use, and which norm, and God knows what. You know, and here it's all just specified and documented. And here's a little bit of coding. Basically, this plugin just calls the dependency. So it's 180 lines, and that's very easy to to get this into the GUI and all that. And that's why actually there's so many methods in the toolbox, because it's very little coding overhead to make some. And the associated prediction function for this machine learning method, the second function, is probably even shorter. So it's this here, which outputs the predictions. It also calls some function of the some dependency in C++ or so, I guess. And I show you the last plugin category that's interesting, and that's the paradigms. So I'll show you the windowed means paradigm. These things are in the paradigms folder. They are all called paradigms and then something. Windowed means it's a class. So, but it's an M file, some documentation, and that's it. And there's 3 or 4 functions defined. The first one defines default signal processing parameters. Right? A paradigm includes how to filter things, among others, and here's where it's defined. And then it has a function to adapt features. This one doesn't adapt features. It just, it's hard-coded. You type it in and you're done. And a function to extract features. And in this case, it's some reshaping and whatever, because it's ultimately just averaging. So feature extraction is a one-liner. And the last one is the visualization function. This is what brought our plot up. It's optional. So if you're really being parsimonious, you can write a plugin without all this, without all the documentation. I just prove a point. So basically, this can be your BCI paradigm. And this will work in the GUI, basically. And obviously, you can go on and do much more advanced things here. It's also not really utilizing all the features. There's others which do much more stuff. In particular, there's some which, I just bring this up, which don't have a separate feature extraction and machine learning part. They do it all in one step. And so here's one which uses a lower level function other than feature extract and feature adapt to do things. So there's some facilities in the toolbox that allow you to write these kinds of things as well if you want the flexibility. Yeah, and so that basically concludes the scripting overview. So we've covered the GUIs, the scripts, some of the plugins. Obviously, there's a lot of stuff to discover, but there's no way that I can really go over all these things in the short time that we have. So I think that's sort of the end of this little demo.